there, let's get started.
Okay.
I'm delighted to welcome Margaux Steyaert, who is here to discuss her PhD project. Margaux is in the Department of Zoology, and she's working on benthic diversity across coral reef structures, genetics, and photoanalysis, and in the Chagos Archipelago. I'll let you explain all that, Margaux. Go ahead.
No problem. Yeah, hi, hopefully you can hear me and see my slides. So yeah, my name is Margaux Steyaert. I'm a third-year PhD student at the University of Oxford, as well as the Zoological Society in London. And today I want to introduce you to some of the work that my lab group and my collaborators have been doing, some of our research goals and some of our early results, as well as my own PhD project. So all of our work is based across studying remote atolls in the Indian Ocean. Specifically, we study an area of the world called the Chagos Archipelago. And this is where we work specifically is a marine protected area, so MPA for short. And this is situated just south of the Maldives, as you can see here. And so why do we focus on studying coral reefs in such a remote place? Well, a marine protected area was set up there in 2010. And with fishing being illegal and pollution being very low, it provides a unique opportunity to study reef ecology, as well as the effects of climate change in a system where the reefs are near pristine and human impacts are somewhat removed. So as you can see on the map on the right-hand side, where the landmass and islands are colored in black and the atoll waters are colored in blue, whilst we have very little landmass, we have a lot of reef and banks to study. So currently there are 66 scientists across 22 academic institutions that study the reefs there as part of the marine science program. Some of the work covered by these teams range from ocean physics and chemistry to reef diversity and productivity, as well as the movement and behavior of large animals like sharks, turtles, and seabirds. If you're interested in learning more about what's been learned from this MPA in the last 10 years, I would invite you to come and read our latest paper that was released at the end of last year. And this was written by everybody who's been involved in the marine science program in the last couple of years. So first, how are the reefs doing in the Chagos Archipelago MPA? Although they are remote and somewhat protected from human impacts, these reefs are also experiencing increasingly frequent spikes in temperature. Here, for example, is a graph showing a reconstruction of sea surface temperature in the Chagos Archipelago across time. As you can see, spikes in temperatures are increasing as in frequency as we get closer to the present day on the right side of the x-axis. So this is something we're seeing across the world. We're seeing more coral reefs getting warmer, and that's leading to quite critical bleaching events and other stress on corals. In fact, severe bleaching and mortality of corals happened in 2015 and 2016 in the MPA when there was a global bleaching event. The graphs here represent the 4 major atolls in the MPA, and the colors represent the number of maximum degree heating weeks. So essentially, the 2015 heat wave caused 60% of shallow water corals there to bleach and die, following quite a long bleaching period. And then the following year, in 2016, 68% of the corals that did make it through 2015 bleached again, and that caused an even further loss of corals. So this back-to-back mass bleaching event in the Chagos Archipelago was really devastating, and it kind of sets up the scene for our project. So overall, the aim of my lab group and collaborators were all funded by the same grant, and that runs from 2018 to the end of next year. Our aim is to understand how reef biodiversity, specifically that of benthic and plankton communities, as well as environmental drivers, which includes physical and chemical factors, and genetic connectivity all interact together to enable reef recovery and production. So I'd like to briefly introduce everybody who works on this team first, and then eventually lead you to my own PhD project and try and explain how it fits into this web of marine research. So first, there are 3 people that work on coral connectivity at the moment. That includes Laura from Oxford, Luigi from ZSL, and Catherine, who supervises and leads this project, along with Chris Jessen at the ZSL Institute of Zoology and at Oxford as well. So Catherine and Luigi work on understanding how colonies of different species of corals are connected genetically across reef systems and atolls in the MPA. They focus on 3 of the most common species. That includes a tabular acropora, a branching acropora, and a massive smooth brightes species. These, like I said, are some of the most common that we see on the reef, but there are also some of the corals that were hit the hardest by the bleaching events in 2015 and 16. So, as well as analysing corals from the MPA, Catherine has been collecting samples across the Indian Ocean, especially on the east coast of Africa, to look at how reef connectivity works on a wider scale in the ocean. And then working on the biodiversity in the MPA is myself and Rosie. Rosie is based at the Imperial College in London, as well as ZSL. So she just started her PhD this year. And on our expedition coming up in a month, she'll be sampling the water column at various sites across the atoll to try and collect both environmental and community DNA to look at both reef diversity and plankton diversity. And then there's me. So my work focuses on collecting and analysing samples from the reef benthos. So I'm particularly interested in studying the diversity of the invertebrates that are associated with the reef matrix. I personally work quite closely with our Stanford collaborators, and they work on the physical and chemical properties of the reef systems across the MPA. So as you can see, my project sits somewhere between biodiversity and environmental drivers. I'm going to start introducing my own project now, and eventually I'll explain as to how it links in with Stanford's work. So like I said, I study the diversity of invertebrate reef organisms, and especially those that live within the hidden parts of the reef. And these are sometimes called the cryptofauna. So what are cryptofauna exactly? They are basically characterized by the small, the hidden, and the encrusting organisms that live in and on the coral matrix. That includes sessile organisms, so organisms that are attached to the reef. That includes sponges, tunicates, bryozoans, and cnidarians, as well as mobile critters that move around. That includes crustaceans, worms, and mollusks, amongst many, many others. And although they're mostly inconspicuous, they're hard to notice, they actually represent the majority of metazoan reef diversity and are really understudied in comparison to the more conspicuous groups on the reef like fish and hard corals. But they play really vital roles in reef functioning by acting as filter feeders, detritus eaters, and importantly, as prey for a really large portion of reef fish. So whilst they're underrepresented in traditional survey studies, new genetic tools and sampling methods and protocols are starting to allow us to study them in a lot more detail than previously possible. So one of the important aspects of my PhD was to try and figure out a way of sampling these organisms from the reef in a non-destructive manner. So like I said, this is a protected area, and in order to look at the cryptic diversity, which is often associated with live and dead coral, it would be quite destructive to sample from the natural habitat. So in order to minimize the impacts of this project and to try and standardize my sampling, I use what we call artificial reef monitoring structures, aka ARMS. And these are made of 9 stacked PVC plates with gaps in between them, and overall serve as a substrate with varying microhabitats for organisms to colonize and grow on. They're gradually being used around the world and are quite popular for cryptofauna studies, and they are actually a pretty ideal sampling and biomonitoring tool for both studying cryptofauna, but also the microbes that are associated with them. So how do you use ARMS to sample reefs? It's quite straightforward. Essentially, you put them together and then you attach the devices directly onto the reef using a lot of bars and a lot of zip ties. And then you leave them fixed and undisturbed for a period of time, and that can range between 1 to 3 years, sometimes slightly longer. And simply during this time, sessile organisms will start to colonize and grow onto the plates. And that in turn will provide a home and attract mobile species. So different studies have analyzed ARMS data differently, but a lot of them follow similar protocols that use both DNA and photoanalysis to identify the collected specimens. So I personally collect a whole range of samples using ARMS devices, and that includes photos of the recruitment plates, as well as photos of large individual specimens that are bigger than 2 millimeters. And then I also look at the DNA side of things using individual and bulk tissue samples from the sessile organisms attached to the plates, as well as individual and bulk samples of the motile organisms. And I divide them between using sizes into different fractions. And importantly, I also take transect line benthic videos of the surrounding arms, the surrounding coral cover around the arms in order to get an idea of what the local benthos looks like. So for example, here is a picture of an arms device in the forefront. As you can see, it's starting to camouflage slowly into the reef matrix. And behind is a set of instruments that our collaborators from Stanford use. They use those to record local environmental conditions. A little bit about that later on. But firstly, I'm going to introduce my personal sampling strategy for my project. And in 2018, 27 arms devices were deployed across the MPA, across 3 different sites. This includes one lagoon site called Ile de Coin, so at the south of the Paros-Banos atoll. We've also got one very exposed reef in an island called Moresby, so that's on the top of the Paros-Banos atoll. So an ocean-facing reef, and then another ocean-facing, slightly more sheltered site at Ile Anglaise, which is in the Salomon atoll on the bottom picture. So I did collect 9 arms, so 3 from each location in 2019. And whilst last year was made impossible because of the pandemic, the idea is now to go back out in a month's time, if all goes to plan, and again next year in order to continue building a time series. So overall in my PhD, I'm hoping to investigate a range of questions, including how the invertebrate community grows and builds over time. So I'm quite interested in succession, but also how environmental conditions and factors interact with the benthic community structure. Actually, one of the most important goals of this project first, though, is to provide an in-depth analysis of what actually is on the reefs in the Chagos Archipelago. We have very little information when it comes to cryptic benthic communities, and so far, no one has used DNA to look at them in this particular part of the world. So like I mentioned, one of the important aspects is the collaboration with Stanford. And what they do is they try and understand the local physical and chemical conditions at the same sites as the arms devices. So here's a close-up of the Stanford instruments. This whole setup is called a beams device, and that's capable of measuring a whole range of factors, including current velocity, wave action, temperature, oxygen, pH, light, salinity. There's a lot of things that they measure. And usually they leave these instruments out for a couple of months whilst we're on expedition. And all of these different parts are able to record data every minute, and in some cases, every couple of seconds. So this whole system enables the team to calculate both the net production and the net calcification rates of the reef by analyzing both the dissolved oxygen in the water, as well as the reef alkalinity, so the pH. So they also put out long-term temperature and oxygen loggers on or near the sites of the arms. And that's in order to look at seasonal patterns and trends of the temperature of the water within the reef, not just on the surface. So the idea here is that by collecting both long and short-term environmental data at the same time as collecting the arms devices, that we can start getting a really refined look at both the physical and chemical world on the reef, as well as the biological benthic communities. So how do we carry out this work? Our research takes place usually in March, April of every year, because that's a weather window where sailing is possible. And we work and live aboard the Biot patrol vessel. So usually we do this for several weeks at a time. And then during the day, we use smaller crafts to get around our sampling sites. So a typical day on board for me includes an early morning dive to retrieve one of the arms devices or carry out other work for collaborators, such as collecting coral samples or doing benthic transect surveys. And then the rest of my day is usually spent processing the various specimens that we collect. So we often need quite a lot of space when sorting out all these specimens. And so I often end up outside on deck. I can't complain about the view because it is beautiful, but we do have to get pretty creative with brooms and tarps in order to keep ourselves in the shade. So this isn't, an oceanography research vessel. It wasn't built for that purpose. It was a rig, like a tug boat for oil rigs in the North Sea. So it's a hardy ship, but not always perfectly designed for science work. So like I said, I didn't manage to get any data last year in 2020, but I'd like to go through now some of the results I have from DNA and photos that I collected in 2019. Before I do that though, I just want to sort of very briefly explain how I use DNA to identify specimens. And so for the larger organisms, so the ones that are bigger than 2 millimeters, we carry out what we call standard barcoding. And that involves extracting DNA from individual specimens and then amplifying a very particular gene region of interest using PCR. And this is also called a barcode. And then this small piece of DNA will enable us to identify individual specimens using a traditional sequencing platform like Sanger. And for the bulk samples of both the motile organisms and the sessile organisms that we take, we carry out what we call meta-barcoding. So that's where we simultaneously extract and amplify barcode regions from all of the specimens in the samples, and then amplify using PCR. In this case, I'm quite interested in the COY and the 18S genes. These are great for looking at eukaryotic diversity specifically. And then I use a high throughput sequencing platform like MySeq, and essentially then take that data into a bioinformatics pipeline where I filter and merge DNA reads, assign taxonomy, and then to try and produce a resulting diversity table. At the moment, I'm using a pipeline that assigns amplicon sequence variants, so ASVs, as taxonomic units for downstream alpha and beta diversity. But I'm now looking to try and cluster those into MOTUs. And if anyone else does meta-barcoding on this call, it would be great to carry on that conversation, because I'm constantly trying to learn what's happening in the field of meta-barcoding. So let's look at some of the results from that meta-barcoding. Meta-barcoding the 18S gene region shows me that actually there's the presence of many, many taxonomic groups. That includes macrofauna, meiofauna, and sessile invertebrate groups. So the graph that we have here in front of us is shows the transformed abundance of reads for each of those phyla that you see listed in the legend on the right. And that's for each of the 3 sampling sites. So as you can see, we have quite a lot of reads that are assigned to annelid worms, as well as arthropods, and a series of algal groups and sponges. And with the 18S gene, we recover the presence of just over 6,000 unique taxonomic units. In terms of the diversity we recover with this gene region, we see that the sheltered site, so the Ile de Coin site, which is inside the lagoon, whilst the other 2, Moresby and Ile Anglaise, are exposed on the side of the ocean. So the Ile de Coin site has a lower Shannon diversity index than the other 2. And then on the right-hand side, we have a multi-dimensional scaling plot using Bray-Curtis distances. Essentially, we're trying here to discern how dissimilar the communities are between individual samples. So in other words, we're using both the presence and absence, as well as the relative read abundance of each taxonomic unit to see how dissimilar the communities are between samples. So each point on here represents a sample that's either motile or sessile, and then the color represents the sites. I just noticed that the colors aren't the same between the sites, so apologies for that, but if you just keep looking at the legend, it should make more sense. So what we see here on the right-hand side is that the sessile and motile communities of Ile de Coin in green cluster together, and they actually cluster away from the other sites significantly, and that the 2 exposed reef sites are actually more similar to each other, even though they're from different atolls. So whilst I was in the field, aside from taking community DNA, I was also collecting and photographing over 750 individual motile specimens that were bigger than 2 millimeters. So what we see from looking at the numbers of these is that there's a clear difference in between the different sites, with, again, the lagoon sites, Ile de Coin, having much less macrofauna than the other 2 more ocean-exposed sites. So most of the specimens that I sampled included decapods, so shrimps, crabs, hermit crabs, and squat lobsters, as well as mollusks. But we also found quite a few cryptic fish species, some juvenile sea cucumbers, some brittle stars, some sea spiders, and lots of other quirky things. I'm actually in the middle of barcoding these at the moment with a series of COI primers. And I'm really hoping that managing to get this finished will help me to refine a species or taxa list for these arms sites, but also try to help to build a local database of DNA amplicons that can be used in the future. So aside from taking DNA samples, I was also taking high definition images of the arms plates whilst on board the vessel. Having these pictures enable me to look in detail at the recruitment success on each plate. So basically, I want to know how much of the plastic square is taken up by live organisms or not after a single year, and if so, by which functional groups. So there are several microhabitats across the arms devices, like I mentioned earlier, and I'm quite interested in seeing how the recruitment and composition of sessile critters differs between them. So for example, between each plastic plate, we have a gap of a couple centimeters, and some of these are what we call open gaps, and that's where the water can freely flow through. And then we have closed gaps where we have plastic spaces that essentially break off that space into 4 different partitions. Then we have the plate faces, so either the top surface or the bottom surface of each of the plastic squares. And I'm also quite interested in seeing how the sessile communities look between the top and bottom of plates. So in order to do this and to analyze these plates, I've been using an online software called CoralNets. And using this, I've been assigning 225 points per plate photo randomly, and then identifying whatever lies underneath that point using a set of labels. And you can see the labels in the middle here. They're used by, and they were created by NOAA's Coral Reef Ecosystem Program called CREP. And these represent the major functional groups that we see on coral reef benthos. So in January, when the UK went into a full lockdown, I decided that it was time to tackle this data set. And I ended up analyzing all 153 plate photos. So we have 17 plate faces across individual arms units, and we've got 9 arms. And in total, that ended up being just under 34,500 point identifications. So a lot of time spent on my computer, but worth it because we now have quite a big data set to work with. Something else I ended up doing as I was going through these was keeping a note and account of all of the hard coral recruits I noticed. So as I went through the photos, I saw that these were popping up only on certain parts of the arms devices and on certain sites only. And I was quite interested to see if there were going to be overall patterns of recruitment that differed between the sites. So I kept a note of these and highlighted them every time I found them. So some results from the plate photo analysis. So first, let's look at the recruitment success. In the table here, we have the summary of the percentage of space across the arms plates that had successful recruitment by organisms. So the amount of space where live organisms were found to be living on when I was doing the photo analysis. We see that Ile de Coin, which is again this lagoon protected site, has the lowest recruitment percentage across the 3 sites, but that has the highest instances of bite marks, so that can come from fish or grazing invertebrates, but also the highest amount of sediment. I was also then starting to be quite interested in how the recruitment success might differ across a single arms unit. So what I did, as you can see on the top right, is that I labeled all the individual plates from 1 to 9 in a descending order. And here is, I'm going to explain all the graphs. And here's the results for the recruitment success across the top and the bottom of plates across those arms as you go down. So basically on the x-axis, we have plate 1 going to plate 9, 3 graphs for 3 sites. And then on the y-axis, we have the percentage recruitment on available space. And what I've done is that I've split the top and bottom of each of the plates so that we can see the differing patterns. And what we do see is that indeed there are some signals of as you descend through the arms unit, as you go from plate 1 to plate 9, we see that especially on the top side of plates, recruitment drops off the closer you get to the reef benthos. So a possible explanation for that is that the closest we get to the ground, the more sediment we see sitting on the top of plates, and therefore maybe the less stuff can recruit and grow. Interestingly, we don't see this on the right-hand side in Ile Anglaise as much, but we do see it in Moresby. So it's not just a case of seeing this trend in the lagoonal site where we expect more sediment to be building up, but we're also seeing it in an exposed reef where sediment is not very present, basically. So then I wanted to look at the composition of the communities, either on the top or bottom face of each plate. As you can see, I was starting to get an idea that there was quite a lot of difference between both these plate faces. So for example, here we have Ile de Coin, our lagoon site. the x-axis are the different functional groups that we assigned points to. And then on the y-axis are the counts of those particular functional groups per plate. And then we've got, for each of the functional groups, we have the 3 arms replicates that are found at Ildecoin. So what we see, and these are just visualizations, but we still get pretty strong signals of patterns, is that the recruitment is very different on whether you're looking at the top or the bottom of the plate. So for example, we see a lot of soft worms living on the top side of the plate and very, very few living on the bottom of plates. But we see a lot of encrusting and calcifying algae living on the bottom side of plates instead of the top. Similarly, we see sponges and tunicates, which are filter feeding organisms, living on the underside of plates and again, not on the top side of plates. So I did the same plots for the other 2 sites, and they were quite similar to each other. In comparison to Ildecoin here, in the other exposed sites, there was a lot more calcifying organisms found on the plates, such as bryozoans, bivalves, and calcifying algae. But what about the main calcifier on reefs, sclerotinian corals? So interestingly, and in contrast to all the other trends that we've seen so far using the photos and the DNA, is that we see a really strong difference in hard coral recruitment across sites. What we see here is that actually it's within the lagoon that we're seeing all of the young corals pop up, and very, very little, if none, on the exposed reefs. So it's interesting because corals are synthetic organisms, and you expect them to be sitting on the top of plates in order to be facing the light. But instead here, when we look at the Ildecoin column, we see that the majority of those recruits were actually found just on the bottom side of plates. So an interesting pattern to note. I'd love to discuss this with anyone if anybody has any theories. My personal one is that with, again, sediment lying on the top of plates, maybe that's stopping young recruits or larvae from attaching. But what this is showing us is that within a single year, we knew that a year before there was nothing on these plates, that there's a very healthy number of very small coral recruits settling on arms devices. And it kind of proves that corals are still breeding in this area, which is nice, and in itself, potentially a positive sign of recovery. So quickly, I just want to summarize some of my own PhD findings so far. So firstly, using DNA, we recover a really highly diverse community using both the 18S and the COI gene regions from what is essentially 7 meters square of colonizable reef space. When you put that into context, I think that's about 2 king-size beds. So not a lot of space, but a lot of data from those arms devices. I didn't mention it earlier, but the COI gene data set I'm still working on, and there's a whole lot of data in there, but I'm reworking through it. So that's why I didn't present it today. And then the sessile and motile communities are actually very similar within sites and within arms replicates, but vary really differently between exposed and sheltered sites. And finally, we see that there's less macrofauna and lower diversity overall within the sheltered lagoon site compared to the exposed reefs. And then using the photos, we see that actually recruitment does differ within a single arms unit, especially between the top and the bottom of plates, and in some cases, as we move closer to the reef benthos. We also see that there's a higher recruitment of calcifying organisms on seaward reefs, so on the ocean exposed sites, compared to within the lagoon. And finally, coral recruitment seems to go against that trend and is observed within the lagoon, but barely on any ocean-facing reefs. So this is my set of results, and I'm now currently working with Stanford to look at their data, and we've got some pretty exciting indications that potentially calcification rates are higher on the exposed sites, which would be, mirroring what I'm seeing here. But we're writing that up as a paper now, so hopefully I can publish that in a couple of months. So I just want to say thank you for having me today. I welcome any questions either now or by email, and I want to say thank you to my supervisors, collaborators, and of course, field team. Thank you.
Thanks, Margaux. That's wonderful. What an amazing set of data set that you've been analyzing over the past few months.
Thanks.
Can I encourage everyone to put their camera on, or some of you to put your camera on for the discussion? And we don't have any questions in the chat, so I'm assuming you have lots of questions in person. Would anyone like to start with questions or comments?
I can kick off with a couple. Thanks for a really nice talk, really interesting to learn about that. So one of the most striking results you had is there was much more colonization of new coral going on in the protected area. And in my ignorance of marine systems, why would you get much more colonization in a protected area, once you don't have the sediment issue, than in the open?
I mean, my theory, and I mean, I think Bry is on this call as well, and he's a coral specialist, so he might be able to give some more details. But I think my instinct is to think that maybe there's a lot of breeding that's happening inside, a lot of spawning that's happening inside the lagoon, and it's just pretty much down to currents. So the way that if you're on an exposed site, then you have a lot more sort of prevailing currents that probably take those larvae away, and so you probably harder to recruit to swim down to a reef when there's a lot more wave action going on. But yeah, you're right. It's interesting that in a site where there's a lot of sedimentation, we see more corals. And I think that's why we're seeing them just on the bottom edge of the arms plates, where they're still on the outside and they can still see the light, but they're not being hampered by the sediments covering. Bri, do you want to?
Yeah, actually, hi guys. That's really interesting. Excuse my look. I've just come in from a very long run. Which is why I'm looking as athletic as I do. Yeah, do you know, one of the things that we found in certain parts of the reef is that actually, even if there are high sedimentation rates, as long as the corals have a really good energy supply and access to the sun, then they can actually kind of use their mucus production and their polyps to actually remove this sediment. So as long as the reef system is a really healthy one and they have got access to those kind of nutrients, then sedimentation doesn't seem to be an issue. And certainly when you look in other parts of the world, like in Singapore, which have extraordinarily high sedimentation rates, the corals there are at a bit of a risk there of exhaustion, I suppose, physiological exhaustion, because they spend so much time removing the sediment. But of course, sediment levels in the Chagos Archipelago are nothing. I mean, as you saw from Margaux's wonderful photos there, the visibility is incredible, and that's some of the cleanest water on the planet that you saw in those photos. But yeah.
Yeah, I think it's down to probably sediment, but I think it would be interesting to see how many of the corals within the lagoon and on the outside of the lagoon are actually spawning every year. So we tried to figure out the timing of spawning last year with Bri. We were on an expedition which got cancelled because the pandemic started whilst we were out there. But yeah, there's not that much knowledge at the moment about the breeding season. There's a lot of work that happens on hard corals in the MPA that's been led by Anne and Charles Shepherds for many years. But yeah, we're just sort of scratching the surface in terms of coral recruitment. And what I'd like to do is, when we go back out in a month's time, is to use what usually is used for coral recruitment studies, which is UV light. So you can excite the sort of photosynthetic algae which live inside the coral. And I'd like to do that with my arms plates, just to see if we can pick up even smaller coral recruits, because I was doing this just by eye. But still, I was able to count, some coral recruits that were only a couple of millimeters wide. So.
And can I ask, your data sets are, so you installed the arm plates in 2018, you said, but do you have the plots been set up longer term, or is that when the plots were set up, the permanent plots?
No, so that was the day that they were fixed onto the reef for the first time, at least for this project. And now it's going to be, when we go this year, it's going to be, more than 2 years, it's going to be 3 years recruitment, and then it's going to be a whole lot more things on there, and I think it's going to be quite overgrown, so it'll be quite interesting to see what happens. And then again in a year's time it will have been 4 years, so hopefully we'll see lots of interesting things, because I think one of the things that's quite interesting about the arms device is that you're actually, compared to traditional recruitment plates, which are just one plate, we don't get much predation or grazing, because a lot of it is inside these quite tight gaps. So you do get this overgrowing of sponges and algae, and it'll be quite interesting to see which of those taxonomic groups are kind of winning the turf war. I'm quite interested in looking at that this year.
Oh, you have such a robust data set. Mark?
Thank you. Thank you for a model, what I think is a model talk of clarity. Wonderful.
Thank you.
I'm a terrestrial ecologist, as you can probably see from the background to my picture. So it's a practical question. in your arms, do the plates come out individually so you can extract them? And I presume you take them up to the boat, to the ship, to do your analysis, and that works fine, does it?
Yeah, so it's this whole protocol was set up by the Smithsonian and Nancy Knowlton, sort of, I think it was over a decade ago. And the idea is it's really easy to take apart. So you have these plates that you have these bars that go through the plates, and then you basically just stack them in and then stack them off. Sometimes they can get a bit stuck because you have, organisms that are attaching themselves to different plates. So you have to break those off, but no, they're quite easy to separate. And then you basically just have them as individual plates. We do that back on the boat. So what we do is we go down, we put a big bin over the arms when we're diving. We secure that with zip ties and bungee cords to trap all of the organisms that are living in there. Because if you just take up the arms device by itself, all of the motile organisms, the shrimps, the crabs, everything will just get scared and leave, and then you won't get any of those. So we have to trap them in, bring them on the boat, and then keep them in a cool vat of water with some aquarium bubblers, just so that we can slowly process everything up until we put everything in the freezer. So it takes some time to go through everything.
No, thank you. It sounds all sensible, but I suspect it's a lot more work than appears. But thank you.
No worries.
Hi, Margaux. May I ask something now?
Sure.
It is super cool to hear something different to what I do. I do more tropical ecology and forest, and getting to know more about this is fascinating. I was wondering, I work a lot with remote sensing and spectral data and structural data. And I was wondering to what extent people have been doing that in your site as well, doing some structural analysis. So how different are these reefs, let's say in the places that are more safeguard in comparison to the others that are more in the open sites? And to what extent this structural complexity and these differences in the structure may explain also some differences in the diversity patterns that you see between the sites?
Yeah, so one of the important things that Stanford do is they measure all the environmental factors at different parts of the reef, so on exposed reefs and inside reefs. And one of the things they do is they look at rugosity, so how complex the reef benthos is in a sort of a 3D structure, and how that affects the way that waves and currents and tidal, water movement move through the reef matrix. So what they're doing at the moment is they took thousands of pictures and they're putting together like a 3D model of the reef in different parts. And that's part of their analysis of, right, okay, so how much wave do we get? And, these are right next to where we're taking these arms devices. So do we get a lot of current? And in that current, do we get a lot of oxygen? Do we get a lot of light? And, all those individual measurements of sometimes I think what's lacking from biological studies. So there's many other studies that have used arms devices to look at benthos. And when they try and do environmental comparisons across maybe a latitudinal gradient or across different sites that are protected and non-protected, the environmental data used for those usually come from satellite data or loggers that take data every couple of hours or every couple of days. And so what we have here is incredibly specific environmental data sets and ocean physics and ocean chemistry at the same time as doing a biological project. So I don't have a lot of replicates, 9 arms devices isn't a lot compared to other arms-based studies. But what we do have is this really specific look that's combining physics and chemistry as well as the biological side of things. So we're hoping that's kind of a nice way to merge 2 fields in oceanography that usually don't even interact. Very rare that like ocean physicists and chemists talk to biologists. So
cool, thanks.
No
I think it should be, Margaux's been very humble here, but she hasn't overstated the fact that this is an extraordinarily difficult site to get to. In one of the most remote coral reef systems in the world. And we essentially have to rely upon a lot of military help to kind of get out to where we go. And they're not, I mean, they help, of course, but I mean, it's not in their job description to drop everything to help us. So I think what Margaux's done, she talks about her limited data set, but it's incredible that she has what she has, given how hard it is to get to the site that we get. And as she mentioned, we were caught out there by the pandemic last year in something of a romantic adventure trying to, when every territory bordering the Indian Ocean had closed their borders to us. So it was, yeah, I think it shouldn't be overstated how much has gone into what Margaux's actually got and pulled together here. But I think that's apparent in the talk she gave.
Thanks, Brett. Yeah, it's a bit of a nightmare to go work in the middle of nowhere when you can't get a commercial flight to your sites. So I think if you work in the Maldives, you can get your own flight there and that's fine. But this time we rely on getting military flights or getting let into a military base that usually is quite closed off. So that takes a lot of time and work, definitely. But it's worth it.
Well, I'd say you're probably the last generation that's going to be able to do this because the world gets smaller. So I'd enjoy it and put up with the difficulties. But I agree with Brian. I think you're doing a great job.
Thank you, Mark.
Just to follow up on that question about the biophysical measurements. If I heard you correctly, you were saying that that team derives productivity from the chemistry measurements. How exactly does that work? Is it from the oxygen levels, or how does that?
So from my understanding, and this is led by Alexi, one of the PhD students who just started at Stanford, and what they do is they look at the dissolved oxygen. I don't know if you remember, there was a long tube on the BEAMS devices, and what they do is they look at respiration rates, and with light they also look at sort of photosynthetic productivity or availability, so PAR, like photosynthetic active radiation. And what they do is that they can get what we call NCC and NCP, so net coral production or net coral calcification rates, and look at the ratio between those, and that essentially gives us an idea as to whether the reef is getting better and accreting versus the reef is getting worse and slightly dissolving and maybe being more overgrown by algal groups. And what we see in her preliminary results is that whilst they're both sort of on the up, so they're both accreting reefs, we actually see that within the lagoon, it's slightly, so it's really just on the border of being healthy and not healthy, whilst the exposed reefs have a lot more calcification potential. And that's kind of mirrored in our functional group analysis using the photos, where we see that there's a lot more bryozoans and other organisms that use calcium carbonate in their skeletons, and they're doing better on the exposed reefs. So we're starting to see some nice patterns between their data set and our data set, but it's really difficult, and we're missing sort of the big gap in the middle, which is microbes. So we've got, you know, the benthic community down to meiofauna, and then we're missing this microbial diversity in the middle. And that's something that I am now going to be working with, hopefully in the next year or so, from some of our community sample data.
A related question I had was with the ARMS device, especially after 1 year or two, is your sampling biased towards pioneer type organisms? Are there longer lived organisms that wouldn't colonize an ARMS over 3 years or so?
Yes, I think you're right. I think what I was already seeing are empty bryozoan colonies. So for example, bryozoans they're kind of like lace, called moss animals, and they grow really, really quickly and they invade lots of things quickly, but then they die off quite quickly as well. And so what I'm seeing is lots of these like lacy skeletons that aren't colonized by living organisms anymore, but instead there's now sponges that are recruiting on top of that because it's a nice thing to grab onto. So I definitely think that it's a shame we couldn't get any data last year because I can't see what happens in the second year after recruitment. But I think that, yes, I think it will definitely change. And there's definitely, you can see wars between different sponges and different ascidians where, you know, there's a lot of sort of competitive interaction where they're sort of trying to fight each other off, and they've got lots of toxins. So you see all these borders between sponges all sort of trying to fight for space and things. And it's really quite interesting. And I wish I understood more about this world. I'm just starting to get a little peek, but yeah, it's incredibly complex. And even with 34,500 points, you're still not really scratching the majority of the stuff that's on there. But yeah.
Oh, I have a question, if that's okay. So I did a little bit of stuff looking at settlement of priorities colonies in Bermuda, trying to get them to settle in the lab. And it was the first year, so we just set up the lab, so everything was a bit of a mess. But we were trying to get them to settle onto tiles, and we found that the settlement of these was better if there was encrusting algae on there. I wondered if that was something you noticed with where you found your priorities colony set or your coral colony settling.
Yeah, definitely. So when I looked at where all these sort of baby corals were actually sat on, it was always some kind of brown encrusting algae or CCA, so like sort of the pink calcifying algae. I think those, I mean, tell me if I'm wrong, Bri, but I think those have been shown to release some kind of signal that does actually help and sort of guide baby corals to the reef, sort of like, hey, here's a landing spot. Yes, we definitely see encrusting algae around where the baby corals sort of prefer to settle on. But we see more calcifying algae on the exposed reefs where there aren't any corals, but I don't think it's because of the same reason.
Yeah, I mean, Margaux's absolutely right. So, you know, with tiles, so yeah, CCA and their associated microbes produce these kind of pheromones, kind of like perfumes that attract, as Margaux says, like landing lights, essentially on the coral reef that bring these down. And of course, if you're a sessile organism and you're settling there for what potentially could be centuries, you have to be very sure that where you settle is a really good place for you to feed and absorb sunlight. But yeah, Ellen, and one of the things you find, well, certainly my work out in Australia was that that if you use brand new tiles and put them down, you get hardly any recruitment. And what we traditionally used to do was actually just sink our tiles in the ocean for several months to allow these kind of benthic crustose algae to kind of settle on them, which then produces this much more amenable surface for these things to lie on. So yeah, it's a really again, you know, as humans, we stumble into these environments thinking we have a grasp of what's going on, and more often than not, nature proves otherwise. But we're getting there, as Margaux says, and certainly this work is contributing to. I mean, it's again, and to emphasise, you know, the effort we're going here. I mean, this area has been worked on for probably the last 20 or 30 years, and the rough estimate is that we've essentially studied around about 2.5% of the entire archipelago. So it's a huge task, and but again, a task that we'd like to try and meet with open arms, I think.
Definitely. I think for the recruitment tiles as well, we had a really interesting conversation last year because one of our field team members was a coral biologist from Australia, Viv, and she was saying, she looked at the arms and she said, well, that's strange because usually when we do coral recruitment, we put the tiles vertically and not horizontally. And I thought, well, that's really interesting. Why? And she said, well, because then you don't have sediment that sits on there and you get more access to light. And I thought, well, that's quite interesting. What if I did that with my arms? What if I literally just put them on the side? And so last year, whilst we didn't pick up any arms, I did manage to put out some more. And I put half of them flat, like you saw on some of the pictures. And then some of them I just literally put on their sides. And I'm quite interested to see how that might change recruitment, to see maybe if with no sediment sitting on the plates, whether actually it might change things. I don't know, I'm just playing around with arms devices underwater. So we'll see.
Margaux, I had a question also. You were at the Coral Gardeners talk a few weeks ago, a month ago, I think.
Yeah.
And I remember you were happily surprised at their rate of recruitment and at their survival rates. And you mentioned pathogens, and I was wondering if you had any recommendations for them or insights on their work.
Oh, I wouldn't know in terms of coral biology. I mean, I would say that they could talk to Bri if they're interested in pathogens and viruses of corals. I mean, that's literally what you do. But yeah, it was a really interesting talk. I really enjoyed it, and I think it's a really cool concept. And I hope it works out for them, and I hope that they don't get too intense of a bleaching event, because it can be quite difficult to replant corals without like a really solid scientific understanding of where and why and what species. So fingers crossed for them that it really works.
Yeah. I actually missed that talk, so I tried to log on to that talk, and I ended up with the classic kind of spinning disc of death on my browser. So I didn't unfortunately get to join that talk. So I'd love to connect with those guys if that was possible.
Yeah, of course, we have the recording.
Oh, wonderful! Oh, that'd be great. Yeah, I'd love to get access to that if I could.
And then it would be great if you could get in touch with them. And
absolutely, yes.
Do you have any other questions? If not, I think we can wrap up. Thank you, Margaux. That was really wonderful. For a lot of us who work on terrestrial ecology, it's just such a breath of fresh air to see all this work you're doing, and it's so inspiring. And I hope you'll come back. You have, once you publish the paper with Stanford, and tell us about it.
I have to be honest, there's nothing fresh about the air in the middle of the Indian Ocean. It's
Heavy. Yeah.
It makes us dream.
Thank you. And if anybody has any other questions or wants to get in touch by email, feel free to do so. Happy to chat more about anything that we're doing. And
we were working on a new Biodiversity Network website, so please send us a summary of your work and project, and we'll put all that on there.
Yeah, sure. I can definitely do that.
I just have one last question, just out of curiosity, if I may.
Go on. Yeah.
How pricey is to actually rent, or I don't know, these ship to go, I don't know, for the period that you got, the 2 weeks or 3 weeks that you got to do that field work? Do you have any idea how costly it is?
Oh, it's expensive. And it's also because they're a patrol vessel. So the boat that we work on, the rest of the year is in charge of chasing out illegal fishing boats. And so a lot of the time we'll be on they graciously let us on for a month or two every year. But even when we're on the boat, if there's an illegal fishing vessel that's coming through, they just go, put the science down, we're going to go chase out criminals. And so, it's not the most ideal oceanography research boat, because sometimes you might have to just put your work to the side and the real life kicks in. But it's a good place to work, but I think it's quite expensive. And I think if you wanted to learn about costs and things, the best person to talk to is Rachel Jones at ZSL. She's in charge of all the expedition stuff.
There's, we're told one of the caveats of being on this vessel is that at any point we'll be pulled off our science work to go and chase shark finners. And although all the scientists are going, well, we don't want that to happen, there is a thrill of excitement being involved in the chase. And Margaux and I actually were on one of those chases last year. I say chase, it's a chase in the most boring sense at about 8 knots. And several days of slow ambling across an ocean. But it's a chase nonetheless. And I had heard costs, Margaux, and I don't know where these came from, that the boat costs around £12,000 a day to run. So it's not pocket change.
Wow. It's an interesting arrangement because the foundation, Bertarelli, that funds this, also was quite influential in setting up and supporting the MPA, wasn't it? That's an interesting.
Yeah, well, they came in at the last minute and made up the difference that the British government couldn't fund. So yes, there's some spare change there, I think.
I was mentioning to Margaux before we started that I'll be going there next year because of new funding from the Bertarelli to go out. So I'm quite excited listening about all this.
Oh, fantastic. Maybe we should set up a small Bertarelli network at Oxford.
Sounds great. Great for that. So we'll be looking at the terrestrial system and flying drones and collecting leaf traits and things on the terrestrial system.
Are you going to be looking at some of the forest, or sort of some of the tree growth on the islands, or?
Yeah, and how the nutrient cycle. So the birds on the islands, how that affects the nutrient cycle that washes off into the reefs, and how that affects. A bit grey and looking at how that affects the reefs.
That's incredible. I mean, as Margaux will tell you, we see. So, I mean, obviously these reefs, and one of the weirdest things for me getting out there was obviously the fact that you realise that this myth of the desert island is perpetuated. Mm-hmm. You know, that it's a palm tree that's on, and obviously we know these are invasive species. And so every now and then, as Margaux and I will be on the boats, and you'll come across one of these islands. And I remember Margaux, is it the Takamuka tree, Yadvinda? It looks like it's the Indian Ocean version of the oak. It's this big, beautiful round, and of course it's absolutely full of birds, seabirds, because of course the seabirds can't nest on palm fronds. Yes. And it'll be really interesting to hear more about that, Yadvinda, I think.
Yeah, happy to have a chat. It'll be fun.
Absolutely, yeah, that'd be really really good to hear about. Okay, excellent. Wow. Oh, and you'll see how fresh that air is.
Thousands and thousands of seabirds all living together on a very small piece of land. It gets a bit smelly.
Oh, then some.
Yeah, especially when they decide to come onto the boat in the middle of the night.
And coconut crabs stealing everything that isn't tied down.
Yeah. Exactly.
Well, a whole new world.
Yeah.
Okay, I think I'll stop here. And you know where I am when you need me.
Thanks very much for setting that up, guys. A wonderful talk, Margaux.
Thanks.